Thank you so much, Isabel. I appreciate that introduction. And thank you so much to everyone for your attendance today. I really sincerely appreciate this and look forward to discussing GI360 with you. So let's begin. Today we'll be discussing GI360 extensive assessment of the GI microbiome. So we'll go through a presentation, and then as Isabel mentioned, we will be taking Q&A live at the end of the presentation. So doctors' data is currently bridging the gap, the diagnostic gap. As you'll see with GI360, there's a combination of complementary methodologies in one test. Historically, when identifying bacteria, so we're talking about a stool test, and a large portion of that is going to be understanding what your patient's microbiome, what bacteria are growing in their stool, and then clinically applying that to physiology and presentation. So historically, when we are looking to understand the bacteria, historically, this has all been done via culture methodology. So that is going to be in the laboratory growing the bacteria. And that's an excellent piece of information. And GI360 does include that. Recently, with the advent of technology, we see this resurgence of DNA data. So we see this in research, but also in clinical applications. So GI360 is combining that DNA data, so that's going to be that PCR-based molecular testing, and the microbiology testing for the culture. So we're looking at the bacteria via microbiology culture and then also via PCR. So GI360 has both of those tests in one, both of those methodologies in one test. So you will see that on the report. The benefits of the PCR data is that we are able to identify dysbiosis and pathogens, and we'll see how PCR is uniquely fitted for pathogen identification. The other benefit is that with this data, we are able to standardize a profile that tells us about a normal biotic population of the colon and then a dysbiotic population of the colon. So we will see that as it regards to IBS, IBD, and you'll be able to see where your patient lines up along those matrices. GI360 also includes state-of-the-art stool chemistry analysis. These are the important markers such as short-chain fatty acids, inflammatory markers, immunology markers, digestion and absorption. All of that information will be included as well. Another aspect of GI360 that is unique is the susceptibility testing. GI360 actually includes susceptibility testing to bacteria and yeast that have been cultured directly in your patient's sample, and you will have that individualized information. So this is an excellent opportunity for individualized medicine. Putting all these puzzle pieces together really does allow for clinically actionable results. And this does allow for a standardized profile to address the microbiome. So you have all of this data in one test. And we're really excited to bring this to you and bridge that diagnostic gap, because previously many of these tests and methodologies would be separate, yet they are all included in the GI360. One of the key points of GI360 is what's called the Dysbiosis Index. Now, you will see that we are looking for about 45 different bacterial analytes that have been shown to distinguish a healthy gut, that's the normobiotic or eubiotic gut, from the dysbiotic gut that is associated with IBS, IBD, obesity, diabetes. Now, the important thing about this we're going to discuss is that the methodology is laboratory validated. And in this regard, this is a highly standardized, validated, and CE marked stool test. So that means when we identify Akkermansia muciniphila as an example, you know that that has been validated to accurately identify Akkermansia muciniphila. So all of that work has been done to the test before it is in your hands and you are going to discuss it with your patients. The technical verification regarding repeatability is 2.6%, and reproducibility is 4.2%. So this is less than 5% across the board. Repeatability means that, as an example, when we run your patient's results multiple times in our lab, that we get the same result. And then reproducibility means if we sent that sample to another lab, let's say in Scandinavia, that they would get the same results. And yes, the coefficient variability, it is less than 5% across the board. So this is a highly reproducible standardized test. And the addition of that CE mark, that is the European equivalent of the FDA, shows that there is that additional validation there. So again, this is a highly reproducible test. That is why this portion of the test is utilized all over the world in clinical and research settings. Furthermore, the Dysbiosis Index has clinical validations. The dysbiosis test defines a normal biotic reference range. With regards to the microbiome, one of the questions that arises is, how can we understand what is healthy? And that's the excellent thing about GI360 is the way that the test was developed. We are looking for bacteria that have been shown to designate in specific populations associated with a normal biotic gut. That's essentially the absence of GI pathology. And then the dysbiotic gut, you have the patients that are diagnosed with IBS and IBD. So the N for this group is over 1,000. And you can see that the test is accurately able to differentiate key microbiome differences. Here we have the information about the dysbiotic group. Only 16% of the healthy bacteria, healthy groups show dysbiosis. And then the 73% had IBS and 74% had IBD. As far as the normal biotic reference range, 84% of those individuals had a healthy gut. And that's the dysbiosis index there. So there are some inter-individual variability there, yet at the end of the day, that 74% of individuals that were dysbiotic with IBD and 73% of individuals that were dysbiotic with IBS. So this is very good information that is clinically applicable in these patient populations. Additionally, you will see that there are over 20 peer-reviewed journal articles about GI360. So these journal articles are able to differentiate the good and the bad dysbioses. This is about, again, the IBS and obesity, like we discussed. So these papers are available for your reference at www.gi360.com. So the neat thing about this test is, yes, you can have it in your clinic and you can work with your patients, and you also can know that this is a published in peer-reviewed articles standard methodology. Okay, so that is the technical background about the GI360. Let's take a look at the GI360 in practice, because this is where you really want to be, where the rubber meets the road. We're taking all of this excellent scientific technical information and we're bringing it into clinical practice and utilizing these data points to improve our patients' health, right, at the end of the day. So here we have the GI360 microbiome abundance and diversity information. This is the first piece of information you will see with GI360. And again, this information here is what we have been speaking about with regards to those technical verifications. So you know that this information is telling you what it is telling you. The normal biotic reference range for the green hexagon is going to be this green hexagon here. This is the normal biotic reference range. So this would be a perfect result if your patient's result, which is the white reb, was superimposed on the normal biotic reference range. So this is where we start. This is a lot of information in this beautiful graphic. And this is also very easy to explain to your patient. You can show them, here is your results. It should be exactly here. So here we see some issues with regards to diversity and dysbiosis. So I will briefly explain how to, a quick tutorial. Again, the white web should be on the normal biotic reference range. If it is the white surface area is approximating what you would expect in this hexagon. That represents healthy abundance. So abundance looks actually okay for this patient because the white surface area is close to what we could see that would represent that hexagon. But there is an issue with diversity. And you can see that diversity could be better just because it's not quite making a hexagon. So the actinobacteria, this phyla is lower. The Firmicutes, that phyla is lower. So now, this is actually a pretty good result. You will see sometime that this white web is very teeny tiny pulled in, and that shows decreased abundance. Other times you will see that this web is making a triangle or a rectangle, and that shows significant issues with diversity. So this is a great place to start. The other excellent thing about GI360 is we start with broad-based information. So here we have the bacteria organized by phyla. We have the 6 main phyla of the bacteria, and you will see this in GI360, but if you're perusing research, you will also see these phyla named as well. So the actinobacteria, Verruca microba, Pteronychides, Proteobacteria, Firmicutes, and Bacteroidetes. So this is the patient's result graphed by phyla. Then we will take it one step further and we will look at the dysbiosis and the diversity indices. These are matrices, these are computed algorithms that have looked at every single bacterial analyte in their specific values and created a score for your patient's result. This is an excellent opportunity, again, for individualized medicine. With regards to dysbiosis, remember we talked about those papers. We're talking about IBS, IBD, obesity. We do not want dysbiosis. Dysbiosis is associated with adverse health outcomes. So the healthy score here would be 1 and 2, 3 is moderate, and then 4 and 5 are dysbiotic. For diversity, the inverse is true. You want a healthy, diverse microbiome. The microbiome is not unlike any other ecological system in that diversity is a marker of health. And so you want the healthy microbiome of 4 or 5, and then a lower score would be 3, 2, and 1 would be the worst diversity score. So with regards to microbiome diversity, this is an excellent piece of information, and you will see that research is really increasing rapidly regarding microbiome diversity and health outcomes. Microbiome diversity is associated with a variety of health outcomes, and it is associated with, again, that IBS, IBD. You also see it associated with metabolic syndrome, cardiovascular disease, Alzheimer's. You can also see it associated with extroverted behavior, anorexia nervosa. There was just a paper that I read regarding individual response to COVID-19. So there's an immune system communication there. And the implication is that because the microbiome is really the foundation, is really associated with so many systemic health outcomes, diversity can really be a key to understanding that. So these are excellent pieces of information here, and you will see these pieces on the profile. Let's take a little bit further look. So again, the dysbiosis index and diversity score, here's some more technical information about it. The markers are based on a verification study of over 1,000 subjects, and the healthy normal dysbiosis would be greater than 3. And then with the follow-up study of over 600 subjects, again, we found that matrices did pan out, that the IBD and IBS populations did have a dysbiotic score, okay? And again, that information is available at www.gi360.com if you want to go into reading those papers. All right, so the next piece of GI360, you will see there will be 3 pages of detailed information. And I want to explain a little bit how to read the profile. With regards to the pages about the specific bacteria, the reference interval is going to be this green area here within reference interval. And then that bacteria has multiple options. It could either be 1 or 2 standard deviations above or below reference, or 2 or 3 above or below. Really, the best result would be if every single bacteria was green right within that reference interval all the way down the page, and we'll see that you'll see what I'm talking about in a little bit. So as an example here we have the important bacterium, fecal bacterium proteiniceae. This is a very healthy bacteria that is associated with positive health outcomes in the human colon. In this case, it is very low. So this is 3 standard deviations below the reference interval. So this patient is going to be at risk for low butyrate production in the colon. They're going to be at risk for inflammation in the colon. So here you can take a look at the detailed finding on pages 2, 3, and 4 of the report. At the same time, rest assured that this information is going to be computed in that dysbiosis index, that diversity score, the web, and the key findings on page 1. So all of that information is calculated and represented right there on that first page. Another example we hear is the Akkermansia muciniphila, and I'm sure you've heard of this bacterium recently. It is the most popular bacterium of the moment. For this individual, their results, the Akkermansia was actually too high, and that's outside the reference interval. Again, this information is represented on page 1. Now, the next piece of information we will talk about is going to be the functional guilds represented on page 1. When you see the key findings on page 1, there will be 2 columns. The first column is going to be this information here. Again, with GI360, there's a lot of bacteria that are included in there. And this is excellent information that it will be computed into those algorithmic scores of dysbiosis and diversity. But we're also going to take it a step further, and we're going to look at specific bacteria that are associated with outcomes. These groups of bacteria are going to be the butyrate-producing bacteria, the gut mucosa protective bacteria. These are the bacteria that are involved in mucosal barrier integrity, and that it may be a precursor to intestinal permeability. We're going to look at the gut intestinal health markers. So that is going to be the fecal bacterium, that healthy bacteria that makes a healthy environment in the colon. And then we're going to look at the balance between the gut barrier protective versus opportunistic bacteria. So you're thinking about the balance, you're thinking about post translocation, things of that nature, and then the pro-inflammatory bacteria. We're going to look specifically at those. And that information will again be on page 1, and we will see that there when we get to page 1. So we're taking key bacteria that are associated with key functions in the human colon, and we're reporting that summarized to you on page 1. This is an excellent place to make a intervention, and we'll see that with some of the cases. So here's what you will see on page 1. This is a snapshot of the key findings. Again, the first group is going to be those butyrate producers. The second group is the gut barrier protective bacteria. The third group is the gut intestinal health markers. The fourth group is the pro-inflammatory. And then the last group is that balance between the gut barrier protective versus opportunistic. And the key is, again, green is indicating within normal limits, and then yellow is indicating outside the reference interval. So if this was your patient's result, you would have the web and those 2 scores, but then you would look and you'd say, okay, the butyrate producing bacteria are too low, and there may be low butyrate, but there may not be low butyrate yet. And the thing to do is to feed those butyrate producers. So we want to feed them soluble fiber. And the next consideration is that the gut barrier protective bacteria are outside the reference range. So that means that mucosal barrier can be compromised. So we want to support that. In that case, it's going to be, again, the soluble fiber, but you're also going to utilize your own protocol to support mucosal barrier integrity, glutamine, marshmallow, aloe, things like that. The gut intestinal health markers are within range for this patient, and the inflammatory bacteria are within range. But we do see that those opportunistic bacteria are too high, and those gut barrier protective bacteria are too low. So we really want to support that gut barrier, and we want to help the patient right here at the key findings. So this is excellent information, and we'll see this in the cases as well. So what we have been discussing so far is the microbiome portion of GI360, but that's not all there is. GI360 also includes PCR and culture for pathogens and flora. So if you want to understand additionally the presence of parasites, bacteria, viruses, yeast, GI360 has that data as well. Now we are going to use the traditional methodology of culture and MALDI-TOF to identify flora and pathogens. This is excellent information because the MALDI-TOF library is essentially open-ended library that is connected worldwide to identify bacteria, and there are new bacteria discovered every year. Additionally, for the yeast, right now there are over 300 species of yeast, of which 95 are candida. So there's a lot of information that can be discovered on the report. Here's an example of the microbiome abundance and diversity information that we have been discussing, and this is the detailed information seen on pages 2, 3, and 4. Recall, this is going to be represented in page 1 in the functional guilds, the web, and the dysbiosis index and the diversity score. But here we have the key findings as an example that Akkermansia is listed here. And we'll see this again in the cases. So we have been discussing PCR data, but the question is often asked, what is the role of PCR and what is the role of culture in detection and analysis of stool pathogens? The great thing is that GI360 is using both. So we're using PCR. The pro of PCR is that this is FDA cleared and validated platform to detect stool pathogens. This is a very sensitive and this is a highly reproducible portion of the test. And this is the identification of the pathogens. The limitation of this test is when you're using a PCR probe, you can only identify what you're looking for. And this is akin to fishing with a bait. So if you went out and you were fishing, you would put a specific lure, specific bait on your fishing hook, and you're looking for just the fish that likes that bait. That is exactly what we're doing with this. So we're looking for Salmonella, Vibrio, Shigella, etc. And that's excellent information. This is FDA cleared, and this is highly reproducible. So here you see this is the pathogen portion of GI360. This is a very sensitive portion of the test, looking directly for these pathogens, virus, pathogenic bacteria, and parasites. The next piece of information we have is that we are going to combine that testing with the culture. So let's take a look at that information. Oh, excuse me. We're going to discuss the parasitology first. So the parasitology is here. The parasitology on GI360 is still the gold standard. So please be aware of that because that question often arises. The parasitology on GI360 is a 3-day OMP that is the expert parasitologist looking under the microscope. This is the gold standard of parasitology testing. So that is included on GI360, and this is a global list, okay? Additionally, there will be other markers. These other markers are going to be looking under the microscope that will help us understand more of the patient's picture. These markers include red blood cells, white blood cells, muscle fibers, vegetable fibers. We're looking at the color and consistency and mucus. And now we will discuss the culture. So GI360 includes an excellent culture portion of the test. This is still the gold standard for microbiology. This is utilizing isolated organisms. Once we have a dysbiotic bacteria or yeast growing in your patient's culture, we can take that isolate and put it in directly for antimicrobial testing. You cannot do that with PCR data. So that is a benefit of culture. And again, this is a unique application. This is akin to fishing with a wide net. So this is you're going in the ocean, you're taking out a large net and you're putting it into the ocean and you're grabbing whatever you can in that environment, in that microenvironment. And that's what culturomics is, because we are culturing in multiple different plate and media for the growth of bacteria and yeast. And then what is grown is reported on the report. Again, we're utilizing the MALDI-TOF library. This is over 1,400 genre and species of bacteria and fungi at this moment. So there's a lot of information on here. And that really is, here's an example of the culture. This is the state-of-the-art culturomics. We do culture for pathogenic bacteria, so you will always see that we are specifically culturing, for example, Salmonella. And then from there, it was individualized to your patient. So another opportunity for individualized medicine. Here you have the imbalanced bacteria. There can be anything from 0 to maybe a dozen bacteria here, and then same thing with yeast. So this is specific to your patient. If there were any dysbiotic bacteria, it would be cultured here as well, and we'll see that on some of the test. Here you have an example where the patient has many imbalanced bacteria and many yeast. So sometimes you will see this as well. And then when a yeast or bacteria has been cultured, we will actually take that yeast and bacteria and put it directly in vitro with the susceptibilities, and we will report that information to you, again, individualized to your patient's result. So the state-of-the-art technology really is the combination of PCR going out and looking for specific pathogens or specific bacteria, and then also utilizing the culture and methodology with the MALDI-TOF and utilizing that information for individualized susceptibility testing, and also including the comprehensive parasitology by microscopy and those broad range of stool chemistry markers. So again, GI360 is including all of these complementary methodologies in one test. One of the questions that arises is regarding which GI360 stool profile do we begin with? I would recommend beginning with the GI360 because, again, it includes all the portions that we have been discussing. And then if you want to retest only specific portions, you can order a smaller portion of the test. So these are meant for reordering, the GI360 Essentials or the GI360 Microbiome. So GI360 Microbiome, let's say everything was good on your patient's report except for that abundance and diversity and dysbiosis, you can just reorder the GI360 Microbiome. Let's say they had an issue with abundance and diversity, but then they also had some dysbiotic bacteria and dysbiotic yeast, but they didn't have any issues with stool chemistries. Short-chain fatty acids were good. There was no issues with inflammation. So just reorder the GI360 Essentials so that you do have options. All right. In the few moments we have left here, let's go through some cases. Our first case is a 23-year-old female, has a history of GI complaints of unknown origin. And I'm sure you've seen these patients in your office, young women that have a good amount of GI complaints historically, and they're kind of just living with it. The gastrointestinal workup was within normal limits, so they did not find anything there. And then the patient is consuming the standard American diet. I know you all are in Canada, but you are probably dealing with some individuals on the standard American diet as well. And we know that that is associated with adverse health outcomes. So let's take a look at the web. I want to start here because this is the first place to look is the web. Remember, this is your patient's result organized by Phyla, the actinobacteria, verruca microba, tourniquetes, proteobacteria, firmicutes, and bacteroidetes. And this information here is your patient's result, is the white web superimposed on the normal biotic reference range, which is that green hexagon there. So this is the first place to start. There's a wealth of information here. For this patient, the first thing I would notice is that that top left-hand corner, actinobacteria, is too low. These bacteria like vegetables, so I would consider the patient's vegetable consumption. This verruca microba is too high. Tourniquetes looks healthy, but the proteobacteria actually is too high as well. See how it's pulled out towards there? So with that, I would think about the standard American diet. I would attribute this to excess fats, excess proteins. So that is the consideration there, is to remediate that diet and consider vegetable consumption. And then with the firmicutes and the bacteroidetes, you can see these are too small pulled in. I would think about, for this individual, their soluble fiber consumption. The gut really does need soluble fiber. That's going to be red kidney beans, lima beans, chickpeas, choose some artichokes, garlics, onions, bananas. So we really need to consider the consumption for that. People are really not consuming enough soluble fiber, especially when they're on the standard American diet. So here I would say abundance is probably a little bit lower. I mean, this is not a good amount of surface area, but then really diversity is off. This is not making a hexagon. So let's take a look at the next piece of information. We have the diverse dysbiosis and diversity indices. The dysbiosis index, remember, the higher dysbiosis index is unhealthy. You do not want the dysbiosis. This is a score of 5, so this is an unhealthy gut. As far as the diversity score goes, you want a healthy, diverse microbiome. This patient has a diversity score of 2, so this is unhealthy gut. So we know there's dysbiosis and there's lack of diversity. Let's take a look at the key findings. Remember, we talked about how the key findings are, these are specific bacteria that are associated with this health outcome. So these butyrate producing bacteria, that first group, these are outside the reference interval. So we do need to feed the butyrate producing bacteria. To feed the butyrate producing bacteria, we want to make sure that individuals are consuming 8 to 15, sometimes up to 20 grams of soluble fiber per day. A quick fact is that, roughly speaking, if someone is consuming the standard American diet, they might be actually only consuming 2 to 3 grams of soluble fiber per day. And they need to be, for this woman, I would say 8 to 15. Men, you can go up to 20. So really nowhere near where they need to be. So this patient needs to feed the butyrate producers with red kidney beans, lima beans, chickpeas, Jerusalem artichokes, garlics, onions, bananas. Hummus is a great option. The gut barrier protected bacteria look healthy. The intestinal health markers are healthy as well, but then the inflammatory bacteria are too high. You need to move away from that standard American diet. That does not do well in the colon. And then the next piece of information we have is going to be independent of what we just discussed, really. So that first piece of information we discussed is going to be about the microbiome. The second column here is going to be any other additional findings on the report. So for this patient, we see that there is lower butyrate. Now that makes sense because the butyrate producing bacteria are lower. So we are going to address this with the soluble fiber. The patient does have low beta-glucuronidase. Higher beta-glucuronidase is really a clinical finding. Lower beta-glucuronidase is actually associated with low diversity, so we should expect that to improve over time. And then we do see a culture Klebsiella and a culture Candida. So that really does help to explain why the patient had such significant GI physiology, with the fact that there is dysbiotic gut, there's low diversity, and then there's these cultured bacteria, Klebsiella and the Candida yeast. So go ahead and address this and treat these, and hopefully the patient's symptoms will improve. Let's take a look at the detailed findings. The detailed findings here for the actinobacteria and the Bacteroidetes and Firmicutes, again, this information is computed and summarized on the first page, but we can see the actinobacteria, actinomycetes, bifidobacterium, these are all very low. So the patient needs more vegetables, soluble fiber, but with this bifidobacterium being low, they're going to be at risk of metabolism issues, vitamin metabolism issues. There's definitely, we want to help this patient here before this gets worse, okay? So these are the detailed findings that you will see pages 2, 3, and 4 of the report. Continuing on, we can see for this patient that the inflammatory bacteria, the Proteobacteria and Escherichia species are too high. Again, that's associated with the standard American diet. So you can see the patient's not feeding the healthy bacteria, and actually the standard American diet is feeding these pro-inflammatory bacteria. And then we do have, again, too high Akkermansia, which is not the best. When we take a look at this patient's stool chemistries, we can see that markers of digestion and absorptions are healthy, inflammation markers are healthy. Immunology is actually a little bit lower, so I would just be considering the patient's status of vitamin A, vitamin D, and zinc. We know that vitamin D deficiency is so common, so that might be another factor. And then for the short-chain fatty acids, that butyrate and total short-chain fatty acids are too low. So we do want to support the patient with the soluble fiber like we talked about. All right. And our second case is a 26-year-old female that has a history of GI disturbances of unknown origin. And this individual has a suspecting intestinal permeability as seen on social media. There's quite a few talks on social media nowadays with TikTok and Instagram about a lot of health topics that people wouldn't be privy to previously, but now people are very interested. So let's take a look at this report. Again, we're going to start with the web. The web is the patient's microbiome right here, superimposed on the normal biotic reference range. So we can say here that, you know, they are lower in the actinobacteria. We want to talk about vegetable consumption. They're lower in that Verruca microba. And Verruca microba, one of the bacteria here is going to be that Akkermansia muciniphila. So I would think about vegetables and then polyphenols, because that Akkermansia really likes polyphenol consumption. So we'd have a discussion about dietary inputs. The next piece of information we have is the dysbiosis index and the diversity score. It's moderate. So let's continue on and get more detailed findings. Well, here we see in the key findings, the butyrate-producing bacteria are too low, the gut barrier protective bacteria are too low, the intestinal health markers are out of range, and I think that the gut barrier protective bacteria are too low compared to the opportunistic. So it's very important that we go ahead and support those butyrate producers with soluble fiber. We want to support the gut barrier protective bacteria. Again, it's going to be soluble fiber, polyphenols, and then your own protocol for the barrier, L-glutamine, marshmallow, aloe, so many things to do there. With the gut intestinal health marker, we also want to think about supporting the soluble fiber, supporting the microbiome, and then the balance between the gut barrier protective versus opportunistic is too high. So we want to intensify those strategies. The second column is going to be additional findings on the report, and we will see that the patient has the C. difficile toxin A and B. So we want to take a look at that. Additionally, there are carbohydrates detected, and this can be due to carbohydrate malabsorption, but this could also be due to intestinal permeability. Now, one connection here, when there is issues with the gut barrier protective bacteria, when there's dysbiosis, and then those gut barrier protective bacteria are compromised, then the mucosal barrier can actually diminish, and that can lead to intestinal permeability. So for this patient, I would consider a follow-up test of serum zonulin to see if they do have intestinal permeability. We also offer fecal zonulin. Now, the low secretory IgA is another finding that there may be diminished mucosal barrier integrity, but then they're going to be at risk for infections. You know, we do see that C. diff, so we want to support that immune response. And then their beta-glucuronidase is too high. So elevated beta-glucuronidase, we do want to address that. We can use calcium D-glucarate, make sure they're having adequate vegetables, adequate soluble fiber, and move away from charred vegetables. I would also think about estrogen, maybe do a HUMAP for the patient as well to see their estrogen status, because if beta-glucuronidase is too high, then estrogens and toxins are available to be recirculated. So let's take a look at the PCR. We do see this C. difficile toxin. So the patient, this would be a consideration for the patient. We want more information about that in terms of history and presentation. And then we do see for the stool chemistries here, again, that secretory IgA is pretty low, and then the beta-glucuronidase is too high. So we want to address that. And then our third case here is going to be a 22-year-old male. Recent history of diarrhea, and then believes these loose stools are due to a stressful internship. All right, let's take a look for this young man. Again, the first piece of information we're going to look at is the web. Here we see that we do have an issue with diversity. Abundance looks okay though, because if it was just pushed up a little bit, it would probably be the same amount of surface area, but this diversity is off. So dysbiosis is not there, but diversity is too low. So we want to support, to increase diversity, we want to take a look at the phyla that are too low, the actinobacteria, bacteroidetes, and Verruca microba. We want to support those with their dietary inputs. Additionally, the proteobacteria is too high, so I would have a talk about moving away from processed food, things like that. Let's take a step further and additional information about the key findings. So the functional yields are all within range, so that's good. But we do see there's some issues with the valerates, the lower beta-glucuronidase, and then a cultured bacteria and a cultured yeast. So we want to treat that. With the Klebsiella, the patient can present with that chief complaint, digestive issues, and loose watery stools. So we definitely want to treat this, and hopefully the patient's symptoms will improve. Let's take a look at the other markers. I will just make a note here of the other marker. There's yeast found under the microscope. This is a common question. Now, when there's yeast found under the microscope, I'm just going to toggle back here, and yeast cultured, that makes sense. because the consideration is that yeast cultured is growing. That's the first place we look for yeast. But then we have a second check under the microscope, and this is looking under the microscope for the cellular structure of yeast. If there's yeast growing, we can consider, yes, that makes sense. Some yeast is growing, some yeast is dead based on the life cycle of yeast. Now, if there is not yeast cultured, again, not this case, but in a different case, if you may have a result where there's not yeast cultured, but you see it under the microscope here, you can consider SIFO, small intestinal fungal overgrowth. In that case, the yeast would be growing in the small intestine, but not in the colon. So we'd only see it under the microscope here. So that's an interesting finding. Other markers are within range here. So we don't see any red blood cells, white blood cells. That's good. And then we are looking at the pathogenic bacteria. These are within range, and the imbalanced bacteria are within range. But then we do see, the Klebsiella and the Providentia bacteria, the Providentia bacteria. I had that mistaken. The Providentia bacteria. So in this case, yes, SIFO. We're thinking SIFO, my bad. So we're thinking potentially SIFO for this patient. So with this case, you cannot utilize the sensitivities because it's already dead. When we do the sensitivities, you'll see here, this is taking the Klebsiella, taking the Providentia, and we're taking it directly in vitro and providing that information for you. So this is exactly what killed it in the lab. When you're finding yeast under the microscope, we cannot do that. So you can use your own yeast protocol regarding SIFO, okay? So let's take a look at the patient's stool chemistries here. These look reasonable. We did see that there is lower beta-glucuronidase associated with the low diversity. So really, the take-home is going to be treating the Klebsiella with the natural or pharmaceutical agents of your choice, as well as the Providentia. So you have uva ursi, ionic and colloidal silver with high sensitivity. We have caprylic acid, uva ursi, ionic and colloidal silver. So those are all good options there. All right. Thank you so much for your attention today. And then if we could go over some questions.